topic of tumor development and progression. So, what you had learned yesterday was really the basics of the cancer cell, how the cancer cells look, their different, their basic properties. And today we'll see how that moves up a little bit further. And I'm trying to keep it simple without getting into the proteins and too much into the genes, but you will still have to know a little bit. So I have included one or two of the genes that are my favorite genes here. So tumor development and progression. What that means is that the cells that were precancerous, they are now pushing forward and they're trying to progress into something bigger and they're trying to develop into a tumor. So, oh, there we go. So, I can't really go ahead without just reminding some of the risk factors, but I won't go through all the risk factors. So today we'll do some of the genetic risk factors, just the genetic component. We're not doing anything else. Then we're looking at the process of the development and progression. What happens to the cell? How do they look like? And then how do we stage the tumors? What are the staging descriptions? What is the formal methodology used to stage it? And then we're going to look at a little bit about cytology. And in cytology, we're looking at one or couple of cells, not couple of cancers that have progressed, looking at slides because that's what you want to do at the end of the day. You want to be able to do image analysis. And particularly, I'm going to use the example of colorectal cancer and what colorectal cancer is, how the tumor develops. So, Pooja, I might have to silence you. Thank you. All right. So let's look at a little bit of the genetic factors, not to have too much of diagrams here, but genetic influences have always been there. Not only are they there just to initiate. Yesterday we talked about initiation, but they're also for development further. And so there are two kinds of genes that I thought that you should know at this point. And one is the tumor suppressor gene. This is the gene that actually prevents uh, tumors. So we have a gene whose job is to prevent overgrowth of cells. And we have proto-oncogenes whose job is to cause cell division. And so we have both of these, and both of them really work in a nice balance. But in the case of proto-oncogenes, if there is a mutation, then that is a little bit more dangerous. And then that proto-oncogene, by mutation, leads the cells to multiply faster and quicker. On the contrary, the tumor suppressor gene, one which is called as retinoblastoma gene, so that's it, the RB gene, the retinoblastoma gene, that on the other hand, that gene is a little bit more forgiving. If there's one mutation in one chromosome, then you're still fine. But if there's mutation in both chromosomes, then that's a problem. Then it gives rise to retinoblastoma in children, which of course now is very much treatable. So proto-oncogenes are more aggressive in the sense that they one mistake and then they will drive cancer, and tumor suppressor genes are a little bit more forgiving. One mutation, still okay. Second mutation, then we have a problem. And so other genetic factors are those that are inherited. So that's your BRCA1 gene, BRCA1 and 2. These are mutations in BRCA1 and 2. The cancer will very likely will happen in the daughter of if there's 50% chances that it will take place in the daughter of a mother who has a BRCA mutation. And so it's kind of important that women screen for them if there has been in the family, that they know if they have BRCA mutation. So that's for breast cancer. APC, people have APC, so I put it down here. APC, this is related to colorectal cancer. So, cancer in the colon and the rectum, so colorectal. And this APC gene, it can be, if it is mutated, okay, if it is mutated, then it's possible that that APC, that mutation will lead to, very likely it will lead to colorectal cancer at a certain age. It takes some time, but it will lead to the cancer. So the APC gene mutation, we find that um, it um, it leads to, for sure, it is the direct result of causing colorectal cancer. And so some of these are important to note. Some people have the APC not mutation, but like a truncation, like a little deletion, like it's half gone. And so that kind of, I'm calling that as a mutation, and that can lead to the colorectal cancer. So overall, there seems to be a very complex relationship, even if there is a hereditary susceptibility that, together with environmental carcinogens, do you or don't you get the cancer? Doesn't one get the cancer? And does it lead to the cancer? And why did it not lead to the cancer if we think that it's supposed to happen? So it's a combination of complex relationship of factors. So um, with that, I just want to talk a little bit about, with this diagram, about because we're going to have colorectal cancer in the end, so I might as well talk about a little bit about what the APC mutation does. And so um, we have found that if a person has the APC mutation, and so this is just a mutation, simple, shown here, and the other factors, like I said, in other factors, lifestyle, etc., together, high fat diet. Okay, what is a high fat diet? A high fat diet is one of animal fat. Okay, I don't know if it's necessarily your tele bhaja um, and all that. I don't know if that's it, but it's mostly of animal fat, and it's not from fish fat either. So it is animal fat, and so that is um, people who have a very high animal fat diet and have obesity together, so they can activate, because of APC mutation. Further, they will activate um, this signaling pathway called as Wnt beta signaling pathway. And that's how it's pronounced, Wnt and beta signaling pathway. And this beta signaling pathway, along with some additional mutations or further changes in the cell, because it is a signaling molecule, and so it leads to further changes, and that will cause a um, mm, a change in the lipid metabolism. It will lead a change into, well, it won't lead to a change, but it will, one of the other symptoms will be a difficult lipid metabolism, and homeostasis of metabolism really goes down. Okay, and the cells, because of this, the cells will be kind of like forced to divide. And I do have a video for you in the end somewhere. The cells are forced to divide. And so all of this, together with other additional, more lipid in synthesis uptake, etc., and more synthesis and more uptake together, will cause these cells to further divide. So we will look at, so when you look at that video, I'd like you to remember that APC is a mutation. The APC makes it, the gene makes a protein. It's going to make a protein, which will not be very useful, but it still makes the protein because it's mutated. And you will see the name beta catenin in that video as well. So might as well listen to this right now. So together, this arrow shows you that the lipid metabolism change, and this arrow is going down. So it shows you that balance, your metabolic balance internally also changes. So let's talk about the steps the tumor development, the steps in cancer, the multiple steps. So sometimes people say there are three major steps, and sometimes they will say that there are like four major steps. And and they can be all deemed together. So the three major steps that I've written here are initiation. Okay, so again, for the development, as we talked about last time also, that there has to be somebody's waiting to be there has to be a the step has to be in sequence. It is a multi-step process. Carcinogenesis means the full process by which it's initiated and developed. So it's all the whole thing kind of together. So it's initiated, anything, everything has to start with something. So initiation and the development together is totality is called as carcinogenesis, how it develops. So so number one, we just said that there has to be some kind of a genetic alteration that took place, but then it has to be associated with signaling pathways. When I keep on saying signaling pathways, what do I mean? Signaling pathways, it is one cell that talks to another. Signaling pathway is one cell tells another cell what to do. So you have blood vessels, let's say, and you have muscle cells around them. The blood cells will tell the muscle cells what to do. And then the blood vessel will dilate. So signaling is like talking, communication. And so these signaling pathways will be disrupted, okay, will be disrupted. Your prime example was just like I told you just now, the the Wnt and beta signaling. If that goes bad because of APC mutation, if that gets dysregulated, if it's dysregulated and signaling continues, then it is going to straight away lead to too much proliferation, and those proliferation cells, those cells that are not supposed to proliferate, they will keep on proliferating, and they will also survive, and they will not differentiate too much. So they will avoid to differentiate, but they will proliferate, and they will keep on surviving. So pretty much, so initiation takes care of that part where you can have all of these things that we talked about yesterday, and bring in that first mistake, start to bring in that first problem, in and so to promote the proliferation unnecessarily where a cell doesn't need it. So this is now kind of initiated. So once it's initiated, now the process really is promotion. So promotion is where they're going to be slowly over time, they're going to be accumulation of these awkward cells. They're going to be accumulation of some of these awkward cells. It's not going to be a very fast process like we tend to think, unless it's a childhood cancer. So it's going to be so epigenetic changes take place. So epigenetic means that they are on top of what happened. There are going to be some small changes to the DNA, and they are environmental variations, and it's not supposed to happen, but maybe a DNA can take a small addition, and it's a kind of like non-specific, but then it just silences a gene. So epigenetic changes can take place where it either aggravates or it silences a gene. And so that takes a while, but all I'm just saying is that there is a slow kind of like accumulation. And so this is the process of promotion. And so there's much more genetic changes that we cannot discuss that leads to some of these similar of these slowly over time, a couple of early changed cells. So they're not even neoplastic, they're like pre-neoplastic. So, and they are refusing to, some of these, they are surviving, and some of these are refusing to die. So they're refusing to, they have already acquired some mechanism by which they keep on, they survive for longer. So they're refusing to die. And so they are inhibiting expansion of these during promotion. They will inhibit apoptosis. They will inhibit apoptosis. What is apoptosis? Everybody, you know that apoptosis is programmed cell death. Very nice way to die. Um, Pardon the expression, but a very neat and clean way to die without harming the whole human being or the whole animal or the whole organism. And so some of these processes will continue to increase their cell proliferation, but they will not die. So all I'm just saying is that they're not growing like crazy. They're not these preneoplastic cells. They're not growing like crazy, but they are kind of like surviving better. And so during this time, these pre-neoplastic cells, they're looking for the environment. They're looking around. They are examining the environment, and they're wondering, well, is the environment trying to get rid of me? Is the environment trying to the environment, these other cells, we'll talk about these other cells, all the neighboring cells here, the blood vessels, are they trying to get rid of me? So there's a constant communication. There's this talk between these cells here, and there's this communication between these cells, and these cells are just trying to survive for some time. And the tumor microenvironment is also thinking like, well, okay, so what what is going on here? Should these cells stay? Should these odd cells stay, or should they not stay? And so there is a communication going on until they start to really progress. So now they start to progress. And so when they start to progress, so this is called as the progression and malignancy. Okay, and I have the same diagram here. So in progression, now the changes in the cell, they are really accumulating various very smart changes, metabolic changes, genetic alterations, slight alterations. And they are trying to acquire, they're trying to, so if there's neighboring cells here, they're trying to tell all the neighboring cells, okay, I need your help. You're going to have to you're going to have to try to help me. So, and so it's trying to convince all the neighboring cells to support its growth. So it is now it's a growth. It has not, it is not, it is a neoplastic cell. Why? Because it has not separated out. It's not gone anywhere. It's still not dividing like crazy, but it is dividing. It is abnormal. The bottom line is that these are abnormal cells, and they start to acquire this invasive property. Invasive means they are trying to acquire properties by which they can move out. So if they can just slip out, that would be great. So they're trying to acquire, but they have not necessarily acquired them yet. And that is called as metastatic potential. So they're trying to break away. So far they're like held together here. They're kind of like held together. They remember basement membrane yesterday? So they're kind of like held together. Maybe there's a basement membrane here, and they're trying to break through that basement membrane. They're trying to dissolve the basement membrane. Then there is all of these materials outside the basement membrane. They're trying to figure out how to digest, how to degrade them. So they are acquiring those properties, but they have not done so yet until it progresses to a point where it has acquired the property and it leaves. And this in the metastasis, they can now leave the location and go to a very distant location. And when they go to a distant location, through the blood cell, so this one, when they go to a distant location, there is no guarantee that it will go and survive the other location. And it's just a chance thing that over time, some of them start to like it. So they go further off and they start to colonize, and they form a colony where they are, and then they grow. So there's no really guarantee that even if they go somewhere through the blood, that they metastasize and then they will survive. But the point is that over time, you give it time, progression goes to metastasis. which is simply means that the cells are going to now invade other tissues. So they will leave and invade other tissues. And that is what's going to cause the death. So the tumor, we saw yesterday that the tumor, if it is held together with the basement membrane, then it's still a tumor. It's not really, it's not spreading out. And that all starts from, so just forming just even the basic tumor, whether it's not gone anywhere, it hasn't penetrated anything, then, it may or may not be totally invasive at that time, but it is developing. So in tumorigenesis, now carcinogenesis, now we're looking at slightly bigger cells, more cells. So now we're developing of a solid tumor. I think I might have used the word solid here somewhere in another slide. So the cells are trying to make a solid tumor. So the cells are now, they are multiplying, they have multiplied, and then they have reached a certain size. So size means the group of cells. So the group of cells have made a certain size, and then they cannot go anywhere else. They're kind of like stuck. And so if you have a lot of cells, so if you look up here, if you have a bunch of cells that are all grouped together, then there's going to be locations where the cell is going to become oxygen deficient. And so this tumor, there's going to be center of that whole cancer mass is going to become oxygen deficient, and we call that as hypoxia. And with hypoxia, the cell now has to wonder, well, if I am going to need to survive, then I need to form in blood vessels, then I need to make blood vessels, and I need to be able to. So the tumor decides, and the tumor then actively tells its neighboring tissue. The tumor actively tells its neighboring blood vessel. If you can see the blood vessel here, it's telling its neighboring blood vessel to make more blood vessels. And so the tumor cells will secrete factors, special factors. When I say factor, that means it's a signaling molecule. So it will start sending out these signaling molecules, and the tumor will force it will figure out how to make those signaling molecules, and it will tell the neighboring blood vessels, endothelial cells, those are blood vessels. So if this is your blood vessel, then on the inside of all your blood vessels and capillaries are these cells, and that is called as your endothelial cell. So these endothelial cells, they have this unique property that they can start to branch. It can start to branch from here, and it can make different blood vessels. So here's one blood vessel. It can start to branch and just can make a tube, and it can branch here and make a tube. And so these cells will start to branch out. So endothelial cells, which belong to the blood vessels, okay, that's the inside here. So they're inside, and that's your blood vessel. So they will promote, therefore, angiogenesis. So blood vessel formation. Again, now you know that endothelial cells are the cells to look for, if you were to look for them under a microscope or a tissue or a section, then you would be looking for branches that take place, and a tubule formation uh, for blood vessels. So pre-existing, so there has to be some pre-existing. It's not going to happen like magic. So it's going to be a pre-existing, and it'll branch out of a pre-existing ones. So new blood vessels. So now we have new blood vessels. The signals have been told, and a new blood vessels forms. And look at this, all the blood vessels are going on are surrounding, and so it forms that crab-like look around those cells. And so then so angiogenesis will need to therefore take place, and we will look at it in a bit more detail. And from there onwards, metastasis will take place. Now, since we're covering invasion and metastasis on another day, which is tomorrow, so we will not talk about it in detail today. But the bottom line is that in metastasis, there has to be, before metastasis can occur, there has to be invasion. What is invasion? Invasion is breaking away from that cell membrane. So there has to be invasion. What does invasion mean? That it will look for breaking away from this membrane, breaking away. I hope you can see this. If you're on your cell phones, I'm sorry, this is just too small. It's not going to work if you're on your cell phone. This is too small a diagram. I didn't think that anybody, I thought everybody has their laptop. So pardon the small diagram, but I have more pictures later on. And so there has to be localized invasion. Localized invasion means just breaking in, one or two breaking in into the blood vessel, a couple of them breaking into the blood vessel. That is invasion. And why is it so important? Look at this. There is a series of cells here. I mean, how does a cancer cell break open from them? So the way that the cancer cell would do that is probably it slips in through the cracks. Okay, it slips in through. So it doesn't break any cells, it just slides in through. And so that's a property. And so that's is the property of invasion. How would it do that? And so once it does that, it goes into the blood vessel, then it just works, goes everywhere through the big blood vessels, the small blood vessels. And once it has gone to the blood vessels, then again, from there, it will decide to come out of the blood vessel, come out of the blood vessel and go into the tissue. So it comes out of the blood vessel and it goes into the new tissue. There is no guarantee that it will work, but many a times it starts to colonize. So the word is colony formation, just like you live in a neighborhood. So colonization, and then it forms another tumor at the distant site. And so let's say it went to the liver. So now there is going to be liver cancer. So it's going to be a liver, but we'll call it as a liver metastasis. So we will, oh, that was my large diagram. So this was my large diagram where I'm supposed to have shown you. So for all of these things to happen, here's the diagram that was covered. See that? It's going in from a blood vessel and it squeezes through. So that's the beautiful thing about metastasis, that the cells have learned the art of squeezing and manipulating their shape to go where they want to go. They're not destroying other cells, they're not breaking other cells, they're manipulating themselves and doing the process. So for all of this, all I need you to remember are those critical traits. Remember, I said that there are these features that the cancer cell acquires, and so that was covered by that figure. So those features that it acquires in order to go through this whole transformation process and to go through these multiple essential steps that are required. And that was, oops, okay, I thought I had that. So it looks like I took away that diagram, those, remember the ones with the 6 features, and then we added some more, and we had a total of 8 to 9 features of critical traits. And so those are all traits. So it is to inhibit self-death, it is to support more proliferation, it is to speak with the neighboring cells, to talk to the neighboring cells, and to get them to support growth. So all those critical traits have to be acquired for this whole process. So we will not do any more invasion and metastasis today. We will continue with what else it is. It's my diagram here. So we will continue with what else the cells do during this thing that we're doing today, during this whole process of tumor development and progression. So in the tumor now, that is a very small tumor. It is trying to develop further. It is trying to grow bigger. It is trying to get larger. It's trying to establish. So here in this diagram that I showed you, so in promotion and progression during these, when it's trying to get bigger and establish itself, so what's going to happen is that there will be a metabolic reprogramming, a metabolic reprogramming um, that would take place. And this metabolic reprogramming simply means that the cells uh, um, will just take up glucose enormous amounts. So the rate of glucose will increase a lot. But it's funny the way that they do it. They will increase a lot, and they will produce lactic acid. The cells will produce lactic acid. Do you know when you produce lactic acid? So, for people like me who don't exercise much, or maybe some people who don't exercise all the time, when you exercise suddenly a lot, what happens? You get this very, very painful muscles, right? You get painful muscles, and it hurts for several days. It really, and then it takes a while. So that is lactic acid that is in your muscles. It's the same thing. What exactly happened when you exercise a lot? You probably were not able, your cells were not able to get enough oxygen. And when they don't get enough oxygen, they produce lactic acid. So, our cells will produce lactic acid. It'll be painful. It'll sit in the muscles. It'll take 4 or 5 days before it can you feel normal. And so that's why you have so much pain when you exercise suddenly. So so what I just told you is that lactic acid, lactate, same thing, is produced by you when you exercise a lot and your cells do not get oxygen. But in tumors, they will take up they will take up glucose, all right, a lot. And there is a lot of oxygen, so there is a lot of oxygen. Then why are they making lactic acid? So that is kind of like extremely awkward. They're producing lactic acid, so there is going to be a lot of oxygen, but they are behaving as if there's not much oxygen. So the cells will still take glucose, they will break down the glucose, they will still get the energy, but they will continuously produce lactic acid. This is called as the Warburg effect. This is the Warburg effect from Warburg, who suggested who observed this many years ago. And so the cancer cells, the tumor, is trying to adjust. It's really hardening itself like. So the tumor is saying that I will metabolic means, glucose utilization, amino acid breakdown. This is all metabolic. Okay. Making more proteins. That's all metabolic. So the tumor decides during this promotion and progression stage that it will take up a lot of glucose, sure, but there's a lot of oxygen, but I'm not going to bother. I'm going to produce lactic acid, whereas I could have just simply produced carbon dioxide and just, respired it off. So I could have produced just carbon dioxide. Instead, I will accumulate the cell. When I say I mean the tumors is going to accumulate lactic acid. That is extremely, extremely weird. That is very, very different from normal. So, so in these cells, and I'm just explaining what Warburg saw, but in these we now understand that that's still taking place. So what is happening is that the cancer cells, they are breaking down glucose by a process called as glycolysis. And they are not really going to use the oxygen, and they're not going to They're not going to use all the adequate oxygen that it has. And so typically glycolysis, I'm not going to go into the cell biology of it, but the bottom line is that we call this as aerobic glycolysis. You have a lot of oxygen. It could have broken down the glucose and produced more energy, but it decided to produce less energy and use less oxygen. But the cell has decided to use the glucose. So a lot of glucose is being consumed. And so there's the big weird thing is that why does the cancer cell do it? Why does the cancer cell do this thing? Why do these tumors? What is the advantage to this tumor to further uh, to to further for its betterment and for its own growth? Why is it trying to do that? Why is the tumor trying to do that? Okay, so we call this as aerobic glycolysis, and the question is that is it cancer specific? Well, it seems to be kind of, yes, that normal cells don't like to do that. So if the cells normally if your cells have oxygen and your muscle cells get all the oxygen, they will break down the glucose. They will get enough um, energy in the form of ATP. I'm sure you've heard of ATP, and they will produce carbon dioxide. And your cells, it'll go out through the lungs. And so, for this, why that it should do this and why it should not have, why it should accumulate lactic acid and not have enough ATP, but use all that, all use all that glucose is kind of weird. And so, what we found over the last couple of years that even in spite of having oxygen, this tumor this tumor is not going to use oxygen. So what the tumor is going to do is that the tumor is going to behave like it doesn't need the oxygen or it doesn't have the oxygen. And when that happens, then we in our systems we have this gene that can get activated. It's called as the hypoxia inducible gene. And so, what is hypoxia? Hypoxia is a condition of is having low oxygen, right? So, when our cells, we have this capabilities that when the cells realize that there's not much oxygen, cells can overexpress this HIF gene, and this HIF gene is the hypoxia inducible factor. And so, obviously, this is going to be made. So these tumor cells will start to overexpress. So the tumor cells now have figured out they are now hijacking your your system, and they're telling the cells to make the HIF genes. Okay, it's like saying fire, fire, and somebody puts, immediately people will start coming and giving you water, or they will try to throw water on the house whenever they listen to the word fire. So it's kind of like the same thing. The cell is saying, "Oh, I don't need oxygen. I don't need oxygen." Okay, and the cell gets confused, and the cell thinks, "Oh, so maybe then I should activate the HIF gene." And so when the HIF gene gets activated, the weird thing is that HIF genes will make proteins that favor cancer. It will make new proteins. This gene will cause the synthesis of new proteins that will favor cancer. What will it do? There you go. It will cause those proteins to be made, to be activated, etc., that will lead to more angiogenesis, so more blood vessels will be formed. So it seems to be by by uh, by using glucose without using oxygen, okay, without using oxygen, by using glucose without using oxygen, and where was it? And by producing lactate, and by producing lactate, it seems that there is some benefit to the cells. So one is blood vessels will now be made, and guess what? cells will refuse to take the signals for apoptosis. Apoptosis means cell death. They will be resistant. And so during this progression phase, cells are, this is all at this point, it's really molecular at this point. It's all proteins and molecular activity. So the apoptosis resistance will take place. Cells will not die. Okay, they'll refuse to die. They will have survival mode on. They will survive more. Their survival genes, we have survival genes. Cells have them. Survival genes will kick in and they will not let them die. Blood vessels are formed, and at this time, it will just uh, make sure that it can break down the glucose and it just takes all the nutrients, etc., that it needs. So it'll make the energy, it will get the energy that it needs. So this is a smart way for the cell, for the tumors to now grow bigger. So small tumors now can grow bigger, and they're behaving like there is no oxygen anywhere, but there is. And the reason why they're behaving that way is so that they can activate this particular gene. So that is the Warburg effect. And so, same thing that I just said. Okay. And so, so what's going to happen because of this Warburg effect now in totality? So you have your glucose uptake. It's about 10 times faster. Okay. So it's 10 times faster in the tumors. So in the tumors, it is utilizing glucose. What is it using all the glucose for? The glucose, it's being used to break down, to give energy. Even it's going to use a lot of glucose to get less energy, but it says fine, it'll just do it. It'll break down that glucose. It needs to break down more glucose to get the same energy, to get same amount of ATPs, and it will do that in the tumors, but that is not happening in the non-cancer cells. So 10 times faster in tumors. And so um, so now because the tumors are not taking up any oxygen, they're not using the oxygen. In glycolysis, we need oxygen. Okay, glycolysis needs oxygen. So sorry, not in glycolysis here, but glucose breakdown. So for glucose breakdown, for efficient breakdown, we do need oxygen. Okay, we don't need it for glycolysis straight away. Um, so the cell's deciding that, okay, I'll just take the half process, and I'll just not use any glucose, so I'll experience hypoxia. But but then they will activate all of that. So because of this, because of not using oxygen, like I said, it produces less ATP. All right. So it's breaking down more glucose. It's this weird. It is just saying that I'll use this broken car, and I will use more glucose. Sorry, I'll use more diesel, and I'll use more petrol, gasoline, and I'll drive less. Very inefficient way. So I'll make less ATP. Less ATP means, okay, energy. This is for energy and for all the work. But if I need more energy, I'll just break down more glucose. So these tumors are going to need a lot of glucose to make just the energy that it needs. Okay, so it's a very inefficient way to make energy by using hordes of glucose. So it's like your car that you never got serviced, your car that you never fixed, and it's giving out a lot of side lactic acid, it's giving a lot of smoke, and it is giving you less mileage, and it's taking up a lot of petrol. So that's how it is. And so another mechanism to explain is probably that the number of mitochondria in the cells, mitochondria is the powerhouse. All right, that's probably also less. And it's less because they're not being used. The mitochondria only is used when there's oxygen, but if it's not going to take the oxygen, then the mitochondria are just not enough. there's no there's not much need. So less mitochondria. So this cell, these tumors are hijacking the internal system, and they are utilizing it for their own purpose. And they're still growing. So again, um, it is using a lot of glucose, but it's not growing like crazy. It is maintaining itself. Okay. And so this is just extra information that they produce some they can produce some different enzymes, okay, that kind of like uh, helps it to continue breaking down the glycolysis without oxygen. So breaking down of glucose without oxygen. All right. So in the process, so this is one of the major so I'm going back here, and this is one of the major ways by which this whole process of progression takes place. It does not necessarily lead to metastasis, but progression means simply that it needs to survive better, it needs to figure out how to hijack the system, and it needs to make blood vessels. All right, so let me see. I think we are going to talk about the blood vessels very soon. So, angiogenesis. So, let's see now. This is just a picture again to remind you and to show you the whole process. So, where do you think that there is progression and, sorry, where do you think that there's initiation, promotion, and progression? So, promotion and progression are all typically in this phase here. So, these, so promotion and tumor progression are these two phases. So typically, this is to remind you again that there is a basement membrane. So this is from yesterday. There's a basement membrane. Here's your blood vessel. Here's your lymphatic system. And in dysplasia, this is totally precancerous. There, it has not broken the basement membrane yet, but it is trying to work it. It is trying to go down to the basement membrane. And so this is your last line of defense, these cells. Okay. And so it still has not broken the basement membrane here in carcinoma in C2. In C2 means it is still in location, it is in place, it's localized, and it has not been able to go anywhere yet. And it is, so in this particular stage, these two together, this is the progression. And during this progression, finally, they will be able to break and invade. So this is very close to metastatic metastasis now. So this is like the beginning of metastasis. And then it goes into the blood cells, it's going into the lymph vessels, as you can see here. So this whole so another couple of things that have to happen for promotion, okay, before we go into looking at the cells, is that there has to be these factors, these proteins that have to be secreted. And so some of these are called as the MMPs. So some things have to be secreted, MMPs, secreted by the tumor cells. So that's another part of your promotion. MMPs, metalloproteinases. So they are, these are here. These MMPs are typically, they are here, but they're not really doing anything. So this is the MMPs, they're here, they're not doing anything. And so what these cells are going to do, these tumor cells will either activate the MMPs, or make they will forcefully make the MMPs, and so there will be more MMPs. And what they will do is they will break down this area. So this area, which is, it looks like clean, but it is not easy to break. There's a lot of fibers in here. And so all of these fibers that are in here, this metalloproteases, it will start to break it. So it'll break some here, it'll break some here, it'll break down some more, such that the cells can now break loose. So that is your secreted by the tumor cells. So the tumor cells are actively secreting things that can break this basement membrane, and it can break others, those fibers that are very close by. All right. And so blood vessels, we said, and then even to be able to go through I let's just take keep out the lymph factors. Alrighty, so here it is again, that same picture, just to show you that the basement membrane, there is all of those fibers. Actually, there's all of those fibers are here in this basement membrane, and at this point, the cells are secreting substance to degrade the protein, secreting subs to break away. So what you learned here is that in this slide is that the MMPs, okay, MMPs, they are degrading. They are degrading. So a part of its promotion is that the MMPs are going to degrade the the extracellular matrix, the extracellular matrix, okay, ECM fibers. Also, it's going to degrade the ECM fibers, also degrading the, where did it go? The basement membrane. So it's degrading the basement membrane. The basement membrane is a ECM. It's what was holding everything together in C2. Without that, without this basement membrane, um, no way to hold anything together. So the basement membrane is a member of the extracellular matrix, and MMPs are being secreted to remove all that. Okay. So now that we've come this far, there are staging systems. There are systems that describe the stage of the tumor. So let's say now that the tumor has kind of like has grown bigger and has invaded tissues. So we have to stage them. Is it stage one, two, three, four, as you have heard of? Okay. And how is that done? That staging system is done by the TNM system. So, sorry, the TNM system. The TNM system is used for staging this. And so what it defines is that it looks at various factors, T and M. So where is the totality of the factors is where is the tumor located in the body? Okay. All right. So not every tumor can be staged this way, but where is it located? That's one question. So T depends on the size of the tumor. What is the size of the tumor? How big is it? How many millimeters is it? And these have been clinically decided. So not to our for our discussion today, but they have been clinically decided what is the size of the tumor. And then has any of the cells, has they've gone into the nearby lymph nodes? And that is important. So if it has gone into any of the lymph nodes, so the N stands for N here, nodes. And where is the lymph nodes, everyone? The lymph vessels are close by. The blood vessels are close by. Okay, not shown here, but there everything is close by. So if it enters the lymph nodes, then the lymph node is a place for immune cells, your immunity. All the immune cells live there. So that's going to be a problem. And then has the cancer spread to different parts of the body? Is it only spread very nearby, or has it spread really far? And so those are the three kind of like questions. Okay, so the TNM system is extremely widely used, and we're looking at it in the next one. And the staging system is same for most of the cancers, but only for brain and spinal cord they have a different staging system. So the brain and spinal cord is separated out. Okay. So so now let's look at um let's dig into this a little deeper now. So the TNM system, sorry, is size and extent of the main tumor. Okay, size and extent. How big is it? What is it hitting anything? All right, so extent. Is it pushing against a nerve, etc.? So extent of the main tumor, and so every kind of cancer has its own way of kind of expressing this. And so the main tumor, the first tumor, is called as the primary tumor. Okay. And if it goes to another location, then it is called as the distant tumor. And N refers to the number, okay, number of nearby lymph nodes. Okay, so did it go into the lymph nodes underneath your armpits? And did it go to any other lymph nodes that are in the nearby region? So how many lymph nodes together did it go to? So that's why it is how many lymph nodes. And M refers to metastasis, and in this metastasis, has it spread to other parts of the body and how far? So again, they can be quantified. So it's a thorough process. So look at this. It is look at this. So let's look at the top first. So in the stages, let's just stick to our T, N, and M. So T, N, and M. So let's stick to this process where there are if you have carcinoma in situ, remember carcinoma in situ, that it was still contained, then it's not even called as a it is not called as a T. The T is not there. It's still like a T0. So the T is called as a that is not called as T1. So T1, okay, T1 in terms of the tumor and its size, the size of the tumor, T1 to T4. T1 to T4 are the extent at which the tumor has actually first penetrated. So if it's penetrated the submucosa and mucosa and it is about to go to the, let's say, the muscle, all right, let's, I cannot get into the, uh, this is not the basement membrane, but this is the mucosa, which would be the next, which would be one of the layers. But if it penetrates that, then that is called as the T1. Then if it goes into the next muscle layer, so from the mucosa, let's say there's a muscle layer, okay, before it gets into the blood, that would be T2. All right, then just outside the muscle layer, um, there would be called as the serosa, the subserosa, and then it can go into organs. So T1 to T4 surely has completely the cells have penetrated and left. Okay. Anything before that is all T0. Okay. So T0, there's carcinoma, but it's in C2. So carcinoma in C2 is this one here, is this one here, that you do have a tumor, but hey, it's still inside. So, and and if it's unable to decide whatever is going on, then it can be called as a TX. All right, so carcinoma in situ is TIS. So the lymph nodes, if you can't find out, you're not really sure, then you're unable to access, then it's just an NX. But if the lymph node, there's none of the lymphs have been affected. And so how do you find that? You do that by little biopsies or by examining the lymph nodes and taking little aspirations from there to see if there's anything there. So if there's no lymph node does not have any cancer cells, no abnormal looking cells, then that's fine. That's an N0. And if it is a metastasis, um, if this one is, what's this for? Um, yeah. So there is a movement to different to some nearby. So if there is a spread to the faraway nodes, then it is called as N1. All right, and it's only 1 to 3. But if it's more than 4, all right, further nodes further far away, then it is N2. So maximum is N2. And metastasis is that there is either there is or there isn't. So there's nothing else, So metastasis, either the you find these cancer cells in a distant place or you don't find them. That's it. So it can be M1 and M0. And if you're not sure, then it's MX. So for all of these together, so with T and N and M, so now you can have different permutations and combinations of all of these. All three have different permutations and combinations, and you can now stage it as stage 1, 2, 3, and 4. So the staging 1, 2, 3, and 4 are when they are in T1 and T2. So if you have a stage, then you now stage 1. If you are in stage 1, then there is carcinoma in C2. This is out of the question, okay? So this is stage 0. Carcinoma in C2 is stage 0. So look at this here at the bottom. If you look here at the bottom, then you see that it is T1 and T2. So T1 and T2, this is stage 1. If this is stage 1, then carcinoma in C2 is, as I said, is stage 0. And there is a word called stage 0. Stage 0 means it's not been staged yet, only because there is time. It's you can do surgery and get rid of the tumors. So I have 20 more minutes left. So here it is. So in this staging, T1, T2, T3, T3, T4, I hope you're able to see at the bottom. It's not too small. And so in T1, T2, in stage 1, there's no metastases. So M0, M0, there is no. In stage 2, there is no metastases again, but it's T3, T4. So in stage 2, you have the tumor has penetrated. Metastasis is possible. It has penetrated and it's possible to go through the blood vessel. And it probably has gone through the blood vessel. It has not formed a colony anywhere else. That's it. So in a T4, look at this. In a T4, it has penetrated the other organs. Okay, it has penetrated the other organs, but these organs are nearby and in its own self, and they are in that same organ itself. Okay, they have not gone to a different organ. So again, M0, metastasis has not taken place. Is that okay? So for stage 2. Now let's look at stage 3. In stage 3, there has to be a nodal involvement. There at least 3 to 4 nodes are affected. 3 to 4 lymph nodes are affected. So N1, N2, or um, N1 and N2. So a couple of nodes have been affected. That means it has metastasized, and it has gone to the lymph nodes. And of course, in this situation, it can be any T, any T. It can be any T. If it has gone to the lymph node, at this point, everything is moot. It doesn't matter. It's already gone. So therefore, you can have any T. And in stage 4, it doesn't matter whether it's carcinoma in C2 here or there or anywhere. Who cares? Why? Because it has already gone to M1. It has already gone somewhere else. So if it is metastasized, then it doesn't matter whatever happens, whatever this T1, T2 is. Okay, so at that point, everything is moot. So metastasis is um, is the last is when metastasis is distant, when it is really really distant, very far off, and the cells have started to colonize and you have new tumors. When there are new tumors, then it is called stage 4. So is stage 4 and T4 cancer the same? Okay, well, T4 here, okay, T4, it turns up to be the same in some ways. So T4 and stage 4 are really parts. They're just different ways of explaining it, of this whole classification process, of the whole staging process. So a stage 4 will be called as metastatic cancer. And the reason it's called as stage 4 is because it has already spread, period. Okay. So before that, it's M0 everywhere. So you see that before that it's M0 in stage 3. In stage 3, what is worrisome is that lymph nodes have been affected. And if lymph nodes are affected, again, it can lead to stage 4. So I hope you're all with me so far. And it's the same thing all over again, okay, just to make you understand. So there is a mucosal layer, all right? So here's a mucosal layer. So so this is all like in the smooth muscle layer. So here's your abnormal cells. Here's your abnormal cells in the mucosa. Then there's the submucosa, and then there's a smooth muscle layer, let's say, and the cover of the smooth muscle is the serosa. All right, that's the serosa, and then come the blood vessel, and then come the blood vessel, and along with the blood vessel, there are lymph nodes. So the same thing I'm explaining again. Here's your abnormal cells. Abnormal cells have passed. Forget the basement membrane. It is way past that. So in stage one, it has way past the basement membrane. Um, and so now we're talking about colorectal cancer. The previous picture was also with colorectal cancer, and um, the tumor has spread, but it is still inside the colon. So it's still inside the colon. Okay, and in stage two, it has spread. Okay, it has grown outside the colon, really moved up, but it has not gone to the lymph nodes. So it is very much close to going to the blood vessels. Has it entered the blood vessels? Um, and in stage three, has spread to the lymph nodes, so that it is about to go to the lymph nodes. The lymph nodes have been affected. The blood is also affected here. It's traveling already. But just because it's traveling doesn't mean that immediately there will be cancer on the other side. It takes a while. The cells just don't know how to survive. They are figuring it out. And then the cancer spreads to in the blood vessels at stage four, which has taken a certain amount of time. It goes to a different location and it has spread to a different location. So it's the same diagram explained to you. All right, I'm sorry I'm not able to ask any questions. I'm just moving along. Are you okay? Is everybody with me so far? Because now we're going to move to the next section on colorectal cancer. Are students with me? I'm sorry I can't see you, so it's really hard to decipher. And
Interesting. And I think first semester students, please follow.
I hope they're listening. Okay, so now we go to some histology because we're here to do cytology and histology, right? That's what this workshop is supposed to. So pretty much I try to do that um towards our towards the end of these. So now we're going to take the case study of colorectal cancer in this case, for the sake of cytology, and to tell you how colorectal cancer develops, what does it look like, and um, what are we doing that's normally done, or can we do anything new? And so this is the colon, this is the rectum here, and the colon and rectum. And what happens is that after the age of 50, adults, they will develop, all pretty much many adults at the age of 50, they may develop, a lot of them do develop these things called as polyps. Polyps are like, are the very first stage. They're not cancers, they are like little growths. And so this is no different from, when you age, you have all these warts and stuff on your skin as well. So it's like on the inside. And so these are these mushrooms shaped. Okay, I'm using the word mushroom. So they're like mushroom. Do you see that here? Do you see this here? So there are these polyps. So they're called as polyps, and they're mushroom shaped, and they are on the mucosa. So the mucosa is on the inside. Here's the mucosa, and there are these, well, there are all of these. You see these little uh, these polyps. And so these polyps are mushroom shaped. And so how does it look under a colonoscopy? Not under a endoscopy. It has to be a colonoscopy, only because this would be, let's say, the camera that goes in and looks at it. Okay. Oh, that's the sigmoid scope. There you go. And so this is a polyps here. So that's your polyps. And so you see all of these polyps that are sticking out. And so these are a lot of polyps, and these are fairly big polyps, honestly. And so this would be, the polyps is on the mucous membrane, and this is the first sign. These need to be removed. And so all that the colon, what is it, the gastroenterologist will do is he will take a camera and he will look at it and he will just go and really, in simple language, they will just scrape them off. They just scrape them off. It's like taking a little blade and just cutting them off. Okay, this is fairly big, but these can be all scraped off, just scraped off. And then they will heal over time. So essentially, those mushroom structures, those mushroom like structures, are just cut and removed. Okay, they're just cut and removed by this GI person. And so a lot of people will have a little bit of these polyps. Some have it a little bit, some have a lot, and some people can live with these polyps for many many days. They will. It takes a while. Things take time. And so what does it all end up in? So what you're seeing now here is a bunch of things that happen in the cell, and a bunch of things that take time, and then what they lead to. So let's go look at the top part here. So in the top part, let's see. I think it's easy to explain from this one. So from the bottom here. So you have again. So this is your mucosa layer. So this is your mucosa. Okay. This is just the inside of your GI tract, your small intestine and your large intestine. And so there is some hyperproliferation, some hyperproliferation took place, and you see these tiny little, you see that tiny little growth, a tiny little growth that took place. Okay. And so during this tiny little growth that took place here over time, so this is all benign. And in this GI tract, this little growth is getting a little bit bigger. And so at this point, it is called as a polyps, and it is a adenomatous polyps, adenomatous polyps means that it has its own little gland. It can secrete some stuff. It can secrete things that it wants to. It can tell other cells what it wants to. So it has a little bit of glands. So it's kind of like it is doing things. It is not just some cells. It is secreting some stuff also. But fairly benign. Why? Because it's all contained on that wall. It's all contained in here. Okay. And so this is called as a polyps. And this polyps is, at this point, it can be seen and it's visible. And so this, over time, this abnormal, growth is getting bigger until it looks like a mushroom here. Okay, I'll use this next slide for the bigger picture because it's just easier. That's why I kept it, forgot. And so over time, again, as time passes, it starts to produce extra kind of aggressive cells here. So these are cells, but they're now more aggressive, and they've started to have some different behaviors. Why? Because in a tumor, they can have different kind of behaviors. They can be cells that have different types of behaviors. And so now this is a severe dysplasia. Dysplasia means severe abnormality. And during this time, it will now start to, it is also beginning to secrete things, and it is beginning to grow. So these are the cells now that survive. These are the cells that survive better, and not only will they survive better, they are able to invade the lining from the inside. Remember, this is the lumen. This is where you have the food. So there's not going the other way. So this is the food. This is the GI tract where their food is here. So it is facing the food every day, but it's not going to go that way. It's really going to get bigger, and then it's going to invade on the inside. So now it will invade the mucosa, the submucosa, and then it will go to the blood vessels. Okay. And this is the diagram that is just showing you what's happening here. So on the top, these are the abnormal cells, really abnormal cells that have taken different properties, different roles. They have started to get smart, and they have, so now at this point, it is going to be called as an adenocarcinoma, adenomatous, because it is glandular, it's secreting things, and it is now malignant. Okay, so let's go back to this slide. So what are we trying to explain here at the bottom? So that's the story for up to when you have a polyp. So this one is a polyps here. This is a polyps here. Okay. Um, this polyps, when I call it a polyps, it's really not malignant. So I can't call this because this is malignant. It's already coming out. Okay. These cells are already trying to get released into the bloodstream. So I cannot call it a polyp here. It can be only called a polyp, which is, these are still the polyps, but they look like mushrooms. So I have to now explain the histology here. So in the bottom one, so I want you to think of what happens. I told you about APC gene. APC is a mutation. And what I told you about is a proto-oncogene. Before I said proto-oncogene are those genes that help the cells to divide, but they are not very forgiving. If there is a one mutation in them, and it's only in one chromosome, then one such proto-oncogene RAS will get hyperactive and it will tell the cells to multiply. Don't care who you are, multiply. And so APC is a sorry, RAS here is a RAS is if RAS mutation takes place, then that can happen. And here, these are, I want you to ignore this one, but cell cycle genes, cell death genes, they're all getting affected. Cell cycle genes are going up, and apoptosis proteins are going down. Okay, they're just not functioning anymore. Okay, so I just want you to think of that. So now let's come to the beginning. So what do we have? Look at this. You have a normal colon epithelium. I want you to pay attention here and see that. Do you see how it goes down and then up, down and then up? And these are called as crypts. So in your small and large intestine, you have these crypts. So this is called as a crypt. CRYPT. So a crypt. So you have these scripts. Why? Because you want your food, there's a lot of work to be done. The food needs to be digested. It just gives you a lot of surface area, etc. And uh, the surface area is really at the top. The surface area is really at the top. At the base, okay, at the base, there are these crypts, and these are all very organized looking crypts. Look at them. So there's one, two, three. This is a growing crypt. Four, five, six, seven. Anyway, there are like seven crypts here, very organized, and that's your normal kind of cell. So those are crypts, and then there's a demarcation here. There's a basement membrane kind of a thing here, and then there's probably um connective tissue, maybe here. So what happens is that APC mutation, if it took place over the years, then there will be a small adenoma. A small adenoma. Why is it an adenoma? Because this side is the lumen. This is the GI tract. This is the GI tract here. Okay. It is, if it escapes from, if it escapes from below, only then it is of worrisome. So now look at the crypts. The crypts look very convoluted. All right, they are convoluted crypts. These are good, these are fine, but these are all convoluted crypts. Now come in RAS. RAS gets now mutated. Okay, so it's acquired another mutation. So one mutation here acquired another mutation, and you get a large adenoma. So now we're beyond like polyps. So you get a large adenoma, completely abnormal crypts. Okay, the crypts are not normal until there's it. Now these cells will now force other genes to be activated, and it turns into a full-fledged carcinoma. And how do you know that? Look, it is getting invaded in here slowly. Okay, so this is a full-fledged carcinoma. Okay. And so, in case you didn't understand this, again, it's the same thing, but now I'm going to introduce another word, and that is, and a couple of things. So, it's the same thing, but look at all the um, genes that have to be activated. Look at all the different other genes here, etc., different proteins here. So, let's not get into that. but you do understand that APC is not doing its function. There's a mutation. And so this was the one that I want you to see now. So with all of these acquired mutagens, some hereditary, maybe there's an APC mutation, and then there's some more spontaneous genetic defects. You have developed the first sign is that there is a development of the crypt has become abnormal. And so when the crypt has become abnormal, so this is just to show you that some of these crypts are abnormal. And so let me show you if I have my next picture. Yeah, I do. So this is called as a aberrant crypt foci. So that's the new terminology for you. So all the crypts, the normal, so here you have normal crypts. Crypts are normal here. But the first sign is that you have aberrant crypt. Abnormal. Aberrant means abnormal. Aberrant. Okay, got my spelling wrong. Aberrant. And it's still a crypt. It's aberrant means it's abnormal. And it looks like a focus point. So it's called as aberrant crypt foci. ACF. So an ACF is formed. So that means here, this will first, there will be ACF. So some of these are ACF, aberrant crypt foci. So you are beginning to see, well, this is too late. So some of them are, there will be aberrant crypt foci here. So let's just take a look at this. So new terminologies for you. Aberrant crypt foci is the first sign. All right, this abnormality is the first sign. Okay, this is, so now let me go to the next picture. We'll make it just easy. Oh, I forgot to read up whatever I said here. So this is the mechanism and accumulation of genes defects. So here you have crypts, and you have a normal crypt. See, look at this beautiful normal looking crypt. And this is a human. On the right side was a mouse. I just covered it up. All right, so these are all these crypts have been labeled. They have been stained. All right, they have been stained specifically. So this is very good looking crypts. But next, look at these. These are your aberrant looking crypts. They're all over the place. They don't look like crypts anymore. They are smaller crypts, so it's like all of these smaller crypts are everywhere. And so these are called as aberrant crypt foci because they are focus, so it's called as foci. So aberrant crypt foci. So early visibility of aberrant crypt foci is an excellent way to know what's going on. And so that simply means that if a you're at the age of 49 or 50, and maybe you don't have anything going on, or maybe you're you know, you don't have any full-fledged, it's nothing in your family, then in a colonoscopy, if they take some tissues like this and they simply look at it, then they will get all these aberrant crypt foci. And that, by being able to see an aberrant crypt foci, even in its early stages, this is like too good to be true, but even if it's very early stages, if you can catch that and it can make it automated, I think that would be really, really neat, because that would be able to reduce the burden on the gastroenterologist who does and the pathologist who do these assessments on a regular huge basis. Okay. And so these are heavily aberrant, and then the cells, they are all, coming out from different locations. So this is, the cells are breaking through, but this is still an ACF. So aberrant cryptophyte, this is aberrant. Everything here now, this is like past aberrant. So this is the first sign. These are the first signs. So these are the initial. So if there had to be any detection for colorectal cancer at a tissue level, you're looking for ACF. You're not looking for polyps first. First, you're looking for ACF. Polyps will come in later. So how much? I think I'm almost done. So I will actually go and I'll show this video right now. Um, Okay, my time is up actually, but I will just end it here. Oh, you were looking at the right slide, right? All this time? Okay, anyway. Okay, so this is the GI tract. I've kept the sound muted. Are you able to see the thing?
Yes, ma'am.
All
Yes.
right, so look at this. You're just getting closer, and you have all this villi. These are the your for digestion. These are your normal structures that are um, for digestion. But if you go inside, underneath it, there you go, at the bottom. So that's your villus. That does your digestion, absorption, stuff like that. Now you go down inside and you go to the crypt. And so look at this. There are stem cells. You can ignore that, but um, you can ignore that. But this is the crypt. This is the normal looking crypt, which is at the base. And there are stem cells that are telling it to multiply, and something can go wrong. And so regularly, every 3 to 5 days, you are making a new crypt. All right, you are making new cells like this every 3 to 5 days because of all that work they have to do. They have to be thrown out. The cells have to die, and you're making new cells like that. And they keep on multiplying, and they push upwards. They keep multiplying, and they're pushing upwards. And then they're doing their function, they become villas, they're digesting. So every 5 days onwards, you are making new cells like this. And just to show you that here, those some cells are dying and other cells are being made. And that's really the normal process. All right, so this would be the very normal process by which those cells are being made. They're doing their function and some are dying. And the bottom, the crypt, is still looking nice and healthy and happy. Okay, and I forgot what happens next. So crypts are always every 3 to 4 days. Either this crypt is being formed or another one is being formed. Now what happens is in an APC mutation, how do we have an adenoma formation? That's the video. Again, this is not a histology slide, but it's a good video. And so in an APC mutation, the APC loss means it's mutation. It's not functioning well, or it's completely gone. Um, hold on. See, what did it say? Okay, so anyway, it is APC is lost, and you get these cells they don't have division of labor. These cells are dividing, but before they were blue cells and orange cells, now there's no division of labor. They are just dividing and dividing, but they're not going to do proper work. And then they keep on dividing, and they're dividing more than necessary, and they're not dying. So they'll keep dividing slowly, and some are not dying, and they're looking for new places. And so see how they all get together here, and now you have an aberrant crypt. You have ACF. And when you cut a section of this and you look at it, you have these little, they look like small eyes, um, like small eyes from the top. So that is your aberrant crypt foci. And so I'll go back to my slide. So this is where I was. And so that's so ACF is the first sign. And we still have to do slides. We still have to look at them. And if there's a way to catch ACF even at a very early stage, even a younger stage, then that would be really, really cool by some kind of automated system. And so I know that I'm over time, but I will just take a couple of minutes to talk about which are the proteins. You can't just not talk about proteins. So beta catenin is a very, very common protein that is overexpressed and in these colorectal cancers. And so anything that's brown, so this is a immunohistochemistry. That means you add antibodies You use antibodies, and you use antibodies to beta catenin, because then it'll go and bind to beta catenin. And so I'll just leave that. Just so anywhere that you is brown, anywhere that is brown, that contains beta catenin. And so in an aberrant crop, so let's look at a normal colon here. You don't have too much of beta catenin. Anything that's brown, all that brown location, that's where they have beta catenin. You have beta catenin anywhere that's brown. Okay, but in an aberrant foci, you have too much beta catenin, too much brown. Okay, and this is like huge. This is really ugly. All right, so a lot of beta catenin, and you can just see exactly as I showed you. This is a ACF. This is an ACF, aberrant. It looks like a little eye, and it's an aberrant crypt foci. And so just wanted to show you the histology of this. This is a well-formed aberrant crypt foci, so any person can see this. There's not much of automated thing that is needed here, but at an early stage, I guess it would be. And I just wanted to, I think, finish up with one or two slides that I just wanted to share. And this is from the open source platform called as Kaggle. I would like you to do kaggle.com. I'm making a document of some websites for you so that you can use this later on. So, so just so that you can recall what it is that we've done so far. So this is from Kaggle. I got these diagrams. And so you have a adenocarcinoma versus a benign tissue. So here you have an adenocarcinoma where it has spread to locations. Hard to understand, but they are spreading. Whereas here you can just see that it is contained. So that's why it is a benign tissue. So this one is the benign tissue, and that's the carcinoma. And same for kind of like lung. I think we'll just keep the lung out for today. And I have here a squamous cell carcinoma that is shown here, but let's just ignore the lung and just stick to the. So, so that was my. I think I went over 5 minutes or so. But that is in totality the mechanism. And so, where do you stand in all of this? Where do you stand in all of this? Like I said, is it possible for in progression for you to um, do imaging of cells that are using low oxygen, that they are using, that they have early ACF? Is there such a thing as early ACF? I don't know. So aberrant foci at early. So is it possible to for machine learning to visualize those